Good morning, good afternoon, everyone. My name is James Oxley. I'm an institute scientist here at the Southwest Research Institute. I'd like to spend the next roughly 45 minutes to an hour talking to you about micro and nanoencapsulation and give you a very brief introduction to encapsulation. Before I get into the details of encapsulation, I do want to briefly introduce our institute so you know where this information is coming from, a little bit on our background. So just give me a couple minutes to go through that. We've been around a while. Our institute was founded in 1947. We are a large independent nonprofit contract R&D organization, started out as a 4,000 acre cattle ranch here in San Antonio, Texas, and today we're much bigger. We've got about 1,500 acres, and you can see there's quite a few buildings. Encapsulation doesn't take up all of this. We occupy a handful of buildings in this upper right corner of the Institute, and certainly welcome to come down and visit our Institute anytime. Some very brief operational characteristics. Again, we're nonprofit and independent. That is a unique model that also makes us unbiased towards the work that we do and clients that we work with. All of our work is contract research development, perhaps a little bit on the contract manufacturing side, more for clinical trials and support, not for any finished goods. We've got about 3,200 employees, again, a large campus. Everything we do, especially related to encapsulation, is going to be here in San Antonio, though we do work with clients worldwide, and the Institute's had clients in over 45 countries. Plenty of lab space, approaching about $1 billion in contract revenue last year in 2025. And we have 10 different technical divisions that span a range of deep sea to deep space is one of the taglines that we use. Plenty of patents, though it's not really our business model to leverage those. We're here to work with you and to help you generate your own IP and intellectual property. And we span what we call this valley of death in terms of technology. So we do have attendees today from the universities where you're doing great fundamental science, basic research that hopefully gets published. And on the other end, we have attendees today from industry where you're making your gadgets, your widgets, your lotions and potions on a large scale and probably not doing a lot of fundamental research. So often industry needs to reach back and help solve some new challenges or develop new products. And on the other end, these university labs want to spin out some of their excellent developments and bring them into a product and then production. And that's where we come in. We help bridge those two worlds and really work highly in this applied science space, not only for micro and nano encapsulation, but for a variety of other industries. So getting into what we do here at the Institute. So I mentioned the Institute was founded in 1947. Our first project was 1949. So we do have over 75 years of history in the area of micro and nano encapsulation and controlled release. We have about 50,000 square feet of lab space. So that little spot I showed you in a prior slide, this is our little corner of the Institute. We are opening a new 20,000 square foot clinical supply facility to expand our current GMP capabilities in the pharmaceutical side. Though it's important to note that's only about 25% of our encapsulation work. We do work with food, cosmetics, consumer products, agricultural paints and coatings, all applications of micro and nano encapsulation. We've got about 30 different processes in-house on a lab to pilot scale. So almost everything I'll talk about during this webinar is something that we have in-house or experience with. Again, we have CGMP or good manufacturing practices facilities. We can handle potent hazardous materials, and we also have some biosafety labs as well. So we're really well equipped and broad in terms of what we can apply and how we can help our clients help make capsules, new formulations, and so forth. So hopefully that gives you a little bit of background about our institute. Myself, I've been here 22 years, background as a PhD in chemistry. This has been my entire career, and I'll see myself out through this career as well here at the Institute. And I have plenty of excellent colleagues here as well that can assist. So we've got 5 or 6 different project managers, all with equal or better knowledge than myself as well in many of these aspects of encapsulation. So we're happy to help and assist in any way. So with that, I'll get into the actual meat of the webinar, the introduction to microencapsulation. So it's broken down to roughly a dozen different sections. So I'll give you some history on encapsulation. How did this start? Where did it come from? Definitions are certainly quite important. Terminology, which can be a bit confusing as you start to dive into the vast amount of literature in the area of micro and nano encapsulation. I'll talk about the different types. So it's not just a simple core shell capsule. There's various morphologies associated with that. Of course, give you some examples of products that use micro and nano encapsulation. General characteristics in terms of size, payload, morphology. And hopefully help you answer or begin to answer the question of why should I even microencapsulate, or do you want to even microencapsulate? I'll give you some very high-level overview of the encapsulation processes that are out there, some common materials, and then we'll close out with some information on controlled release, which is a common application of micro and nanoencapsulation, as well as capsule design strategy. So I like to start out by at least roughly describing encapsulation as three parts. So there's art. So these are the technicians that have been in the lab 40 years, scientists that have been in the industry for 40, 50 years, who really have almost an artistic approach to how they make capsules. And they can look at a vial, look at a beaker, look at something that's encapsulated and tell you if it's working or if it's not working without collecting any meaningful data on the measurement side. We don't have time or space to talk about that here. We do have some of those technicians in our lab, but it's our job to apply the science. And so that's the heart of encapsulation. That's certainly what I'm going to talk about. But there is a third component that we can't overlook. So that's the industrial proprietary component. So many of the companies out there, some that are probably on the call today, have their own little niche trade secrets, proprietary components, patented information, which is public, but the proprietary and trade secret information is not. So there may be encapsulated products out there, encapsulated solutions out there, and you're looking to find out how those are made. We may not know, and that's really only known by that company. So, but hopefully, at least the science portion that we'll talk about will give you some insight into perhaps how those products are made. So, getting into the root of what micro and nanoencapsulation is, just breaking down the word itself. So, first talking about the scale, micro and nano. So here we have just a common scale bar going from 0.1 nanometers up to 10 millimeters. Everything from molecular size, proteins, getting into really where micro, or I would say nano encapsulation starts, is the tens to hundreds of nanometer range. And so those can be the size of, I think we're all familiar with the COVID virus or the COVID vaccine, which are roughly actually about the same size, tens to hundreds of nanometers for the vaccine itself. Then you get into micro encapsulation starting around a micron and up. So things about the size of a bacterial cell. Often the analogy is given of human hair that has a width of about 20 microns in size. Very common for a microcapsule size as well. Pollen, it's an enemy of many of us. And you can get larger up to a millimeter in size, maybe large salt crystals, or a nice reference for kind of the largest you might consider for microencapsulation is a period on a 12-point font. That's a couple millimeters in size, and you can start to go larger, but you really start to get maybe into macro encapsulation at that point. So that's the size range. We're operating in this realm of roughly 10 nanometers to about 10 millimeters in size. And then you apply the capsule word. I think we all know what a capsule is. So something that will entrap something within it. So just some macroscopic analogies going from deep sea to deep space capsules, deep sea submersibles. Of course, we've all probably had these gel caps that have pharmaceuticals in them, or you've seen the two-piece capsules that actually have microcapsules within them. But this is the capsule. So you combine those two together, and that's what gives us a microcapsule or nanocapsule. And I'll get some more into the terminology here in just a second. So the history of encapsulation, we can go all the way back to the 1870s. It started with spray drying, mainly to preserve milk and eggs. Get it into a powder form, get the water out of there, give it less opportunity for microbial growth or mold to growth. If you get the moisture out, that helps stabilize it. So a robust and quick and easy way to remove that water was spraying it. So taking the milk, spraying it out of a nozzle into a hot chamber and pulling off that moisture to give you a powder. Now that's not really, we're not purposefully encapsulating at that point, but by default, you are encapsulating the nutrients of those products into a particle. Really where the encapsulation part came is in the 1930s, taking flavor oils, fragrances, which are inherently sticky and liquid and not easily processable as a powder, of course, because they're liquids, but mixing those in with some solid matrix materials and spray drying that to make spray dried flavors. And so that kind of kicked off the early encapsulation era with matrix morphology that I'll discuss here in a second. And then the second big advancement was a core shell capsule. And this was the carbonless copy paper. invention back in the 1950s by the National Cash Register Company here in the US, where they used a gelatin-based coacervation process to encapsulate a leuco dye that when it's exposed to an acidic activator changes color. And so that's what gives you this carbonless copy paper activation. Also in the 50s, that's where our institute began involvement, working a lot with spinning disk atomization, co-extrusion, and drip nozzle type systems. You also have fluid bed coating or Worcester coating, especially work out of Dale Worcester's lab at the University of Wisconsin, which is spun into a company called Coating Place. So we have emulsion base, we have atomization, we have fluid bed coating up through the 1950s, and then you start to get to other emulsion-based formulations. So the 1960s, one of the first publications on what's called interfacial polymerization, basically making the shell between the interface of an oil droplet and aqueous phase, or water droplet and oil phase in the case of this paper from Science. Also in 1966, that's when you start to see in situ polymerization come about. These are polymelamine or polyurea formaldehyde-based shells, in part meant to replace or enhance the gelatin-based capsules that NCR developed. One of the first courses started in 1967. I had the privilege of taking this type of course from Dr. Bob Sparks. I didn't take it in 1967, but that's about the time that he started it. And then one of the first societies emerged in 1973, the Controlled Release Society, still around today, mostly pharma-based, but there is still a nice core group on the consumer diversified product side. And they do have a meeting coming up in Portugal this summer, so look into that if you're interested in attending that meeting. And then finally, in the 1960s or 1970s, even though it wasn't called nanoencapsulation, you have nanoencapsulation being developed. So polymerized micelles, also getting into some polyacrylates and cyanoacrylate-based, would now be called nanoparticles, work done in the 70s. So nanoencapsulation isn't new; it's just become more refined. And then since the 1970s, 50 years have passed, no major foundational processes, rather variations on these themes. So now in fluid bed coating, we have continuous fluid bed coating. Spray drying, we now have electrostatic and nano-based spray drying. And co-extrusion, we now have microfluidic-based systems, vibratory nozzles, emulsion-based processes, advancements in membrane emulsification, simple coacervations, and so forth. And then, of course, nanoencapsulation, without some of its advancements, we wouldn't have some of the vaccines we have today and lipid nanoparticles and so forth. So there's only so much time in the webinar to go through these different variations, but it does give you a sense that really a lot of the basics came about 50 years ago, and then since that time, it's been refinements of these main processes. There is plenty of information out there. So just as an example, using a site like lens.org and searching for these terms, micro, nanoencapsulation, microspheres, almost 250,000 publications since 1950, so that averages out to about 200 a week. You stack that up, that's about the height of the Statue of Liberty or the Atomium over in Brussels. Not enough to squeeze, not enough time here in this 1 hour webinar to squeeze into it, but it does give you a sense that there's plenty out there and likely a solution for a problem that you're seeking. And if not, that's where you reach out to a developer or a research institute to help out. So on the publication side, we also have the field of study side and patents. Almost 500,000 patents and patent applications. So about 275 a week, probably higher than that given the rate of increase. And then I just listed the top 10 that popped up in lens.org. So you can see it's a mix of pharmaceutical companies, consumer product companies, cosmetic companies, academic institutions, and so forth. So it really is broad in terms of who is interested and who is working in the fields of encapsulation, again, dating back to the 1950s. And if you pull out keywords, you begin to see where the backgrounds of these developments come from. So chemical engineering, material science, chemistry, engineering. And then there's some other ones in the background, biomedical engineering, polymerization, porosity, medicine, nanotechnology. So there are some very common physical science and biological science themes behind the fields of study in micro and nanoencapsulation. And then if we break down those publications even further, we see that it's broad and just in terms of sense of the journal. So you can say, well, is there just a journal I can go and read every day, every week that will give me all the information that I need? No, that is not the case. Maybe 20, 30 years ago, but today there's over 300 publications that have, or 157 journals that have over 300 articles that have addressed different types of micro nanoencapsulation in different fields. So chemistry, chemical engineering, material science, pharmaceutics, biology, and so forth. So it's very broad in terms of the sense of where this information comes from. So fortunately, in the emerging world of AI and search engines, it's easier to do strategic searches to find some of the solutions and answers that you're looking for. And it's not just isolated to one country. This is a global technology. So there's over 159 countries that have publications with that show up over 1,000 publications for each of those countries. About a third from the U.S., a quarter from China, probably about a quarter from Europe, and so forth. So it is global. Also representing that, for example, this webinar, we had over 150 people register that range from students to CEOs, universities to Fortune 500 companies, and we have people from at least 3 continents, probably 4, if I dug into some of the companies a little further. So this is a global technology. Getting into the information behind encapsulation, the morphologies, terminologies, and so forth. There's really 2 main types of microencapsulation morphologies. On the left-hand side, we have what's called a matrix particle or microsphere. So this is 1930s spray drying flavors. The little white dots would be your mint oil, your peppermint oil, whatever it may be, and then the matrix would be maybe a starch or something like that. And then on the right-hand side, we have a classic core shell microcapsule. So if I were to tell somebody microencapsulation with little knowledge behind it, they might assume this is what everything looks like. So, the macro scale analogy is here is an M&M. For the matrix particle, my macro scale analogy is a blueberry muffin, where the blueberries are the active, and then down with the core shell, the chocolate is the active. And you can have something in between. So I could put a candy coating around my blueberry muffin, and now I have a capsule inside of a capsule. But these are the main morphologies that you'll find, not only across microencapsulation, but nanoencapsulation as well. And in terms of terminology, there's a few different words you'll find to address the different components, depending on where you learn about encapsulation, what literature you're reading, what era it comes from. So on the top, I'll typically call this a core shell microcapsule. So I'll call the inside the core, but you also may hear it referred to as the IP or internal phase, the active, the payload, the fill or reservoir. And then the outside is the wall, the coating shell, membrane, something like that. On the bottom, again, I'll call this a matrix particle or microsphere. So instead of a shell, it has a protective matrix with all of your encapsulating ingredient kind of bound up within that matrix. And my note to you would be if you're going to talk about encapsulation, just be consistent with how you're describing the different components of the capsule, consistent and accurate, so that you're not flipping between terms. So again, I'll typically call the top core shell and the bottom a matrix particle or microsphere. So for the core shell capsules, it doesn't have to always be spherical. So you can have an aspherical or regularly shaped core shell capsule. That might be the case where you're coating maybe a crystal or a seed or something that's an agglomerate of particles that's not quite spherical. You can have something polynuclear or multi-core. So this is different from a matrix particle. So if you can count the number of core droplets or particles inside of your capsule, that would be a multinuclear. So very, very heterogeneous, kind of on the verge of being core shell. Maybe your core split up into a few different droplets or particles. That's some polynuclear. And then a multi-wall. I think the name is self-explanatory there, having 1, 2, 3, or 4 more walls on the surface of your core shell capsule. Then the other being this matrix particle or microsphere. This is by far the simplest, the easiest to make. So again, spray drying in the 1930s, this was the first kind of true encapsulation form made. Here, instead of a wall, it's a matrix, and this is a continuous matrix throughout. So this is, the muffin of our blueberry muffin, and then the little orange particles are our blueberries. So it's dispersed throughout, typically homogeneously. A disadvantage of this, as you can see, some of these are at the surface. So just like a blueberry muffin, or let's say even a chocolate chip muffin, your chocolate chips are on the surface, and if it gets warm, you're going to get chocolate chips on your hands. So the same is going to be true with encapsulation using this process. You are going to have some, for example, flavor oil at or near the surface, unless you do some post-processing to wash it out. It doesn't have to be heterogeneous. You can have a homogeneous matrix particle. So imagine if your active ingredient is soluble in the matrix. There you could have something that's completely, again, homogeneous. So kind of like a lollipop where you have sugar, flavor, and color all in a nice translucent homogeneous sphere. Very rough idea on cost. We have a whole lecture on this in our workshop. But on the cheap end, often less than $1 or less than €1 per kilo in terms of processing costs. That's where you'll find your matrix particles, maybe some of the polynuclear particles. And this is where you're using atomization, spray drying, melt spray congealing that I'll talk about here shortly, where we're processing large amounts of material through a nozzle up to 10, 20 tons an hour to make capsules. So really it's economy of scale and simplicity that makes it so cheap. You start to move up to a little bit more expensive, some of the core shell technologies, a little bit more difficult, not, I wouldn't say difficult to operate, but a little slower to operate in terms of throughput currently. And then you get into combinations. So I have $3 signs. This could be tens. This could be hundreds of dollars per kilo in processing costs. So you get down to small scale, lab scale, of course, much more than that. And you may need two processes. Maybe you need to do a melt spray congealing process to make the core and then some sort of overcoating to make the shell. And so now you've doubled your processing costs. So it's a very broad orders of magnitude range where you'll find the cost for encapsulation is really case by case and formulation and process dependent. Before I get into the processes, I do want to give you a rough example of what some materials and combinations might look like with regards to the core and the shell that I'm talking about. So I've picked liquids and solids and gases here. So for a non-aqueous oil, so let's say fish oil is a great example. That's been one of the success stories over the past couple decades for encapsulation. One of the best products on the market makes a core shell capsule encapsulating that fish oil with gelatin. So gelatin is dry. It's a fantastic oxygen barrier. You keep the oxygen away from the fish oil. It's less susceptible to oxidation. And giving you those malodor, those really nice oxidized fish smell. Gelatin, of course, coming from either pigs, fish, or cows. So ironically, encapsulating fish oil with fish gelatin, which is a really powerful combination. If I encapsulate aqueous solutions, which on its own is easy, it's the stability and where they're used that is really a challenge with encapsulated aqueous fluids, but it is feasible. Dilute acids being an example. Here you don't want to use gelatin because your core will dissolve your shell. You want to use something that's water insoluble. So waxes or polymer blends, those can be vegetable waxes, candle waxes, petroleum-based mineral waxes, microcrystalline wax, and so forth. And I'll mention a few shell materials later. Solid materials, plenty of examples out there, just roughly pigments maybe used in paints. Maybe you want to put a water-soluble pigment into an aqueous-based paint. So you'll put a good water barrier on there. So ethyl cellulose is an excellent moisture barrier. So derived from cellulose, so coming from either wood pulp or some vegetable sources. And then finally, gases, very rare, but there are a few examples out there. A common one I'll mention a few times is perfluorocarbon gas used in ultrasound diagnostics. And there it's encapsulated typically in a lipid shell to make it biocompatible within the body. And so those lipids coming from either synthesized or modified from vegetable or flour types, sort of like sunflower and so forth, or vegetable oils. So just a few rough combinations to give you a sense of what can be encapsulated. And then we get into some general characteristics. So I've mentioned, we have our morphology, we have our shapes. What about sizes? What about wall thicknesses and payloads? What's possible? What's common? So here I have both listed. What's common is if you go to a commercial manufacturer and say, hey, I want a capsule, make me a capsule. What size can you make? What's possible is where you go to the academic literature. I showed you there's about a quarter million publications out there. You are most likely going to find an answer that you're looking for. Hey, somebody's done this, but have they done it at scale? So the common is more at scale, and there's going to be a few exceptions to this. But in general, if I were to go to a spray drying company, 5-10 microns is most likely the bottom end of where they're going to be. Up to prilling and maybe even fluid bed or some of the vibratory nozzle systems, that's where you can get the millimeters. So, but possible, certainly less than tens of nanometers, especially with molecular complexation. And then 5 millimeters is kind of a random top end. You can certainly go higher, but you start to get into macro encapsulation technology. Wall thickness, this really more applies to your core shell microcapsule. So it's the name implies the thickness of that wall. Roughly a few hundred nanometers is going to be the low end, especially with some of your emulsion-based processes, your in situ polymerization, interfacial polymerization, up to hundreds of microns. As you start to go higher in the thickness of shell, you lose payload, and so it becomes less economically feasible. But you can certainly go higher than 200 microns. It's the low end that can be problematic. Possible, you can have a monoatomic layer. So think about a micelle, just a simple layer of amphiphilic molecules surrounding a droplet inside of maybe an aqueous phase, up to hundreds of microns, maybe even millimeters if you want a really tiny core. And then finally, payload. So this is the amount of active material within your capsule. So the amount of water in the water balloon, the amount of chocolate in the M&M, how much can I stuff in? The amount of blueberries in your blueberry muffin. So with atomization, spray drying, that I'll talk about here shortly, 30 to 40% is more common and maybe on the lower end. You're not going to get much above 50% with a matrix particle. So think about if I try and stuff 50% blueberries into my blueberry muffin. That muffin becomes less effective at protecting those blueberries. For core shell capsules, 95% is a rough high end. As the thinner you go, the more fragile the shell, the less protective properties, the more transmission of oxygen, water, or other outside environmental factors. So 95% is kind of a reasonable top end. 80 to 90 is more common with some of the emulsion-based processes. But what's possible, again, you can go less than 1% and above 99%, but there are massive trade-offs when you get into those extremes. So I mentioned at the beginning, there may be some questions about encapsulation that you should consider before going into this. And the first that we always like to think about when we're talking with clients is, are you sure you want to encapsulate? And it's a true and honest question that you want to make sure you thoroughly answer, because microencapsulation is not cheap. It can be quite expensive depending on what your objectives are and what you're trying to encapsulate. So you want to go through the exercise of, is there another way to do this? So a classic analogy I like to use is a two-part epoxy. So if you go to a hardware store, you can buy those double barrel syringes that have your part A and part B of the epoxy. It's up to you, the consumer, to take that syringe, squeeze them out, mix them together. It gets a little messy. Then you have about 5 minutes to work with it before it really starts to set up. Wouldn't it be great if you could encapsulate one of those parts and put it in the other? When you squeeze it out of the syringe, the capsules break, it mixes for you, and you have your epoxy ready to go. That would be fantastic. Can you encapsulate part A and part B? Yes. Can you put part A or part A and B into one another as a capsule form? Yes. Can you set it up so that they rupture at the right pressure? You're just using hand pressure to break. Yeah, you can probably do that. But will those capsules be robust enough to survive the manufacturing process? That's a little trickier, because you have to be fragile enough to be able to go out of a nozzle and break, but robust enough to survive mixing into the part A or part B in the first place. So it starts to add some complexity to the equation, not to mention the cost of making the capsules and having this product. So suddenly, instead of maybe a $5 or €5 double barrel syringe epoxy system, it may be $25, it may be $50. So then the question is it worth 5 minutes of your time to spend $45 more? Probably not. So that's where packaging might help. But there's certainly plenty of other examples that I'll give you on the next slide. So you just decide, all right, I do need encapsulation. Will that matrix particle work? Will spray drying work? Will melt spray congealing work? These are the easiest and most ubiquitous encapsulation processes. But remember, you have material at or near the surface. So maybe for something like taste masking, you don't want your drug or your nutraceutical at the surface. You're like, well, I can't do a matrix particle. It has to be core shell. Okay, so that decides that problem. Then what size? Where are you putting this capsule? Generally, we recommend what's the largest you can go. The bigger the capsule, typically the easier it's going to be to make. The lower the surface area, the greater the stability, the thicker the shell. There's a lot of advantages going bigger from an encapsulator standpoint of view, but there's a lot of advantages to go smaller from your perspective as somebody who wants to put a capsule in a product. So keep that in, and I'll talk about that a little bit more later. Also, what payload? So how much do you want to stuff into the capsule? What's reasonable? Considering the more you put into it, the less shelly material you have. And then finally, how do you want it to release? So presumably you're encapsulating for a reason. In most cases, it's to release and to protect. And do you want it to release? So think about that ahead of time and how you're going to engineer that into your capsule. So that being said, you've asked that question, do I want to encapsulate? Yes, there's plenty of reasons. So here's just a few. So a tacky liquid to a free-flowing solid. So that's your spray drying example from the 1930s, taking a mint oil, for example, and getting it into a nice powder. Separating reactive components. So that's your carbonless copy paper. That's putting your leuco dye into a protective shell so that when it breaks and releases, reacts with the surrounding environment. protecting from the environment, that's fish oil. You know, it loves to react with oxygen and make some really pungent odors. So if you can encapsulate it, protect it, you've used encapsulation well. And there's others, of course. Moisture can degrade bioactives or biologicals that have been dried, heat, acids, and so forth. Taste and odor masking has seen a great success in enabling children to be more compliant with their medications through chewables, controlling or just timing release. So great advancements, especially in pharmaceuticals and agricultural products, for releasing either active ingredients for your body or limiting runoff of pesticides and fertilizers in a field. More on the pharmaceutical side, but locking in an amorphous or a crystalline state of a drug. And then targeted delivery, if you're not familiar, is used a lot in the pharmaceutical space, but we're seeing more of that in outside other industries. So agricultural, for example, so capsules that target certain pests or certain herbs, or in the cosmetic industry, so more cosmeceuticals that help target certain parts of the skin. So plenty of reasons to encapsulate. And then what can you encapsulate? I gave you a little taste of this earlier. Solids, liquids, and gases. So, you know, all three phases. I'm not aware of plasma being encapsulated, but solids and liquid gases are all out there. There's examples. A few down here. And when asked, can you encapsulate something? The answer is always yes, but there's always a but. You know, the ridiculous example is, can I encapsulate humans? Yes. You know, this is a submersible that was built by the Institute in the 1950s, but it's not going to be micro. So keep in mind, there's always limits and exceptions to what you want to encapsulate, where you want it to go. A few products down here, so solids, whether it be a pharmaceutical or maybe a iron fortification, dyes for a label, or liquid crystals for a label, or again, omega-3s, that's fish oil for edible products. Gases, so if you've had pop rocks, so that's encapsulated carbon dioxide, and then I mentioned the perfluorocarbons earlier for injectables and ultrasonic contrast agents. And there's plenty of applications. So of those that registered for this type of webinar, I think most were represented, and those range from pharmaceuticals, nutraceuticals, flavors, consumer products, you name it. And these pull from one another to develop new products. And we often pull from maybe pharmaceuticals to help develop something new for paints and coatings, or maybe there's something that worked well in agriculture that now will work well for nutraceuticals. So there is a benefit to looking at other industries for some of these applications in encapsulation. Just a few specific examples of not only what the product is, but why it's used and what process was used. And we'll talk about the process here on the next slide. So chewable medication. So if you have kids, now it's great. Almost all the pharmaceuticals that are chewables taste fantastic, and my kids often got them confused with candy, so you really have to hide them. But example is the Tylenol chewables. Fluid bed coatings, one of the methods used to mask acetaminophen. There's also been some use of coacervation. Instant espresso, so spray dried materials. So you're taking a mix of flavors and components from the coffee and getting it into a nice uniform blend that easily dissolves or disperses in water when it comes in contact with hot water. Fabric softeners and fragrance in the fabric softeners has been a very popular topic over the past 10 plus years. So that when you dry your clothes, capsules will adhere to the clothes and then continue to release fragrance throughout the use and wear of that clothing, either through diffusion or friction. So emulsion-based processes used there to encapsulate the fragrances. A lot of skincare and cosmetic products encapsulating cosmeceutical or ingredients beneficial to the skin in liposomes, because they have that hydrophobic nature to them. And often expensive to make. That's why these tiny little bottles can cost quite a bit, but you do see a lot of cosmetic products with liposomes. So that's a nano encapsulation process. Bigger capsules. So I don't think this product's on the market anymore, but this is a seamless gel cap made using a submerged nozzle co-extrusion technology. I'll show here in a second. Encapsulating a mint oil, bite down on it, get a nice burst. And then labeling, so color changing labels with temperature, so have a phase change or liquid crystalline material inside. As it's cold, they're blue. As it's warm, they're gray. So an emulsion based process. Here you don't want the capsules to release. You don't want the dye leaking out everywhere. So just a few examples to give you a sense of not only the breadth of applications, but the different processes behind them. So getting into the technologies, I've divided it into 4 general categories. In the upper left, you have atomization. So this is just spraying something out of a nozzle. So you may have heard terms like spray drying, spray congealing, spray chilling, prilling, spinning disc atomization. There's different types of nozzles, pressure to fluid, rotary. There's newer versions like electrostatic atomization. Great for making matrix particles. Spray coating, so if you're familiar with pan coating, so this is how the M&Ms are coated, or if you're familiar with Jelly Bellies or Skittles, typically a pan coating process. But a microencapsulation or smaller process is fluid bed coating, and I'll talk about that here shortly. Co-extrusion, great for liquid-filled core shell capsules, and there's different variations, vibratory nozzles, submerged nozzles, electrohydrodynamic systems. Microfluidics is one of the bigger advancements in this space, or inkjetting. Or you can even get into classic screw or twin screw extrusion, but that really deviates more from a classic microencapsulation approach, that where we would see liquids instead of extruding thermoplastics. And then the 4th category are emulsions, so or chemical processes, depending on how you want to describe them. And this is where the carbonless copy paper development emerged from. And that's coacervation, but you get into phase separation techniques, in situ or interfacial polymerizations, liposomes, sol gel, and then some nanoencapsulation technology. So if I were to put everything on a slide, this is it. And I'll break these down here in the subsequent slides. Why are there so many techniques? First of all, no single technique can span that entire range that I talked about at the beginning of 10 nanometers to 10 millimeters. Some come close depending on how you emulsify, but in most cases, you're really limited to physics and what's been developed and really scaled up in those processes. So on the top half, you have the physical. So these are your atomization, spray drying, some of the co-extrusion processes, fluid bed coating. Those do great on the larger size range. They struggle once you start to get down into the tens, maybe even single microns. And that's really where you need to transition or think about some of the emulsion-based processes. And now you can start to move down to low microns, submicron, and nanoencapsulation. And these do relate to one another and need to be considered when thinking about capacity of the process, whether it's grams per hour or continuous tons per hour, or something simple like spray drying. On paper, it's simple. You spray out of a nozzle and dry. Or something complex, electrohydrodynamic co-extrusion, where we're applying a high voltage potential to a co-extrusion nozzle to make small core shell capsules. So there's a lot of complexity to that, moving at grams per hour, while spray drying is at tons per hour. And these will move around depending on your application, who you're working with, maybe some special considerations. But for the most part, not a lot on the continuous side, most are on the edge of batch to continuous. And then of course, as capacity decreases, complexity increases, your cost is going to go up. So it's nice to be down here on this lower right end, but not all. Spray drying and disc atomization don't work for everything. So I want to spend just a little bit of time on each of the processes so you have a sense of what they do, why they are called what they are. So atomization, just a very simple cartoon. You're spraying something out of a nozzle. So I think we've all used a can of spray paint, can of deodorant, can of hairspray, whatever it may be. You're atomizing. You are making tiny droplets. Now, what's in those droplets is going to be your capsule. So you either dissolve maybe starch in your flavor oil, or a polymer and an active ingredient, and then or a wax and your active ingredient. But if it's a solvent or water-based, you're going to add some heat to pull those solvents and water out. That will dry it down to your particle. If your liquid is a wax, you spray this into a cold chamber and you pull the heat out. So in spray congealing, you're pulling the heat out, or in solidifying, spray drying, you're putting heat in to evaporate off. You get really nice small particles, single tens of microns. Various commercial equipment available from lab scale, and then of course this is the most production you'll see. So tons per hour in terms of quantity on the larger scale, mainly for something like milk powder. So that's atomization, great for matrix particles. If you want to go to a core shell capsule, that's where we move to spray coating. So you're basically spray painting a particle. So you have to start with a solid particle. This isn't going to work with liquids unless you first encapsulate that liquid into a solid particle. And then we're slowly building up a coating. So a classic is fluid bed coating or Worcester coating, where you're flowing solid particles around in a chamber of air, kind of like a lottery ball or a bingo ball machine, but much smaller particles. As your particles float around, you spray in. So typically from the bottom for something like a Worcester process. the longer you spray, the thicker the coating. And so here's an example in the lower left of some particles coated with a black coating here onto the surface of a white particle. And then laboratory scale equipment that'll handle, tens to hundreds of grams, a little bit larger scale, hundreds of grams, maybe low kilos. And then you can go up some of the largest, you're talking about a ton, maybe a 2-ton per batch, or even some continuous fluid bed coating processes. Move over to liquid filled core shell capsules. This is where you get into co-extrusion. So this is a nozzle inside of a nozzle where you extrude two liquids to form a liquid inside of a liquid droplet, and that outer liquid then hardens. And then depending on what that liquid is, so it could be a melt, a wax, so you're going into a cold chamber and pulling the heat out, or it could be starch or gelatin based, in which case maybe you're adding heat to dry off, or it could be something like alginate, if you're familiar with that, where you're dropping into a calcium chloride bath. The leading edge of this is something like a microfluidic system where we can get down to tens of microns, low hundreds of microns. You can vibrate the system where you can get nice monodispersed capsules, and there are a couple manufacturers out there that make either lab scale and then one or two that make large scale equipment as well. Getting into emulsion-based systems, this is probably the least capital intensive. And as the name implies, we're making an emulsion in most cases. So oil and water is the best example. So fish oil into water, you have gelatin present, condense the gelatin around the oil droplet using what's called a complex coacervation process, and you have your capsule. So in the upper right, beakers, stirrers, temperature control. Scaling up is just larger tanks, larger equipment. But you can also go much smaller now than you can with co-extrusion or fluid bed coating or spray drying. So here we have a tip of a needle injecting with small little, about 200, 500 nanometer size particles made using a solvent evaporation, solvent extraction process. So the emulsion-based processes, we can do matrix or core shell, and it bridges us over to nanoencapsulation. Now, the general definition of nano, arbitrarily 100 nanometers or less, or something having features less than 100 nanometers. For my general take on it is processes that can make capsules less than a micron, because most of the conventional processes I just talked about can't make capsules less than a micron. So you really need a new set of techniques to do that. So that's what I refer to as nanoencapsulation. Plenty of reasons to nanoencapsulate. So one is just to get capsules into places where the microcapsules couldn't go. So drug delivery is a good example of that. A 1 millimeter bead is not going to diffuse well through the body. Higher surface areas, so for better interaction. So imagine encapsulating a flavor you want to release on the tongue. You want a lot of surface area so the water interacts. Maybe some unique optical properties for beverages, or good suspension stability due to that higher interaction of the surrounding environment. Some of the common techniques that fall more into the nanoencapsulation realm, molecular complexation, something off the shelf like a cyclodextrin. So you're complexing a lipophilic molecule with the inner structure of a cyclodextrin molecule. It's not a core shell morphology per se, but you are loosely encapsulating it. Micelles as well. You're making an emulsion with a surfactant. You can cross-link some of those surfactants to make a very thin shell. It may not be mechanically robust, but it can help prevent some coalescence. Solvent evaporation extraction, used a lot in the pharmaceutical space, especially with polymers like PLGA. Lipid systems, so if you're familiar with that COVID, some of the common COVID vaccines used structured lipid nanoparticle systems. You also have liposomes, and inorganic materials. So clays and zeolites can act as some nanoencapsulation constructs. There's a lot more behind the nanoencapsulation space, but this is just a very, again, introductory overview of some of the common techniques. There are some other mentionable methods, something like yeast, so nature's capsule. This is brewer's yeast. So you can either extract out the guts and put something else new in, whether it be flavors, whether it be an active ingredient, some type of oil that will absorb in, or you can engineer the yeast to grow your own and produce its own certain enzymes or proteins or RNA or DNA. Hot melt extrusion is another common one, not the same as co-extrusion. So we're taking our matrix and our payload, something you might use with melt spray congealing, but in this case, maybe a higher melting, passing it through a melting and mixing zone, and you get a nice filament out. Very similar to 3D printing, if you're familiar with kind of benchtop home FDM 3D printing. You take that filament, you can break it up into little rods, or you can grind it down. And so you may not have spherical particles, but now you have matrix particles of your active ingredient within a payload. There's also templating and plating, so taking existing particles and absorbing in an active ingredient, similar to what you might do with yeast. If you want a comparison of processes, so this is a chart I put together a while ago. Different processes, whether or not it forms a microsphere or microcapsule, whether or not the core will be a solid or liquid. I didn't put gases on here, and I won't go through all of these, but this is a good quick chart to refer to, and there is a reference down here. This is a book recently edited by Bob Sobel in the second edition, so one of the chapters has an overview with this table, if you want to come back and refer to it. Getting into some of the materials, we don't have time to cover all the materials. There's hundreds of materials, thousands of combinations, and then once you talk about different variants, maybe tens of thousands beyond that. But very general categories: water-based shell materials, great for encapsulating oils if it's a core shell, or maybe a matrix particle if you want something homogeneous that's water soluble, or heterogeneous if it's oil-based. But proteins, polysaccharides, starches, cellulosics, there's a wide category of modified cellulosics, polyacrylates, latexes, pseudo-latexes, if it's a solvent-borne polymer, and then other hydrocolloids like gums. I think I've said the word melts a couple times. There I'm referring to materials that are typically solid at room temperature and then melt above 50, 60 C. So you can process them at elevated temperature, but will remain solid at kind of standard storage, ambient storage conditions. So waxes, vegetable minerals, so beeswax, candida wax, carnauba wax, a lot of vegetable waxes, microcrystalline wax, even polyethylene glycol, low molecular weight polyethylene can be used. Some solvent-borne polymers, some of the common ones there, especially for biodegradable systems, polylactic acid or polylactic glycolic acid, used a lot in pharmaceuticals. Some of the cellulosics, especially ethylcellulose, is more solvent-borne. And then reactive coatings. So these are going to be shell materials where we're using the chemistry to make the shell. So you're emulsifying into a mixture of monomers to make either polyurea, polyurethanes, polyamides, or formaldehyde polymers, or even some acrylate-based systems. And this is where there's been a lot of recent creativity for making biodegradable shell materials, because that is a big emphasis, given a upcoming ban on microplastics in the EU. Some important properties to think about when you're talking about not only your core material, but your shell material, but your core material as well. Solubility is a big one. Everyone has a different version of what is considered soluble. In the pharmaceutical space, soluble might be, a few mgs per mil. But if you're spray drying, you want something that's highly water soluble, so you can get as much bang for your buck in terms of throughput. So when you're thinking about solubility, think about what the process is going to be and how much needs to dissolve, and how that solubility might change with temperature, pH, and so forth. And the temperature and pH might also impact permeability of your materials, diffusion, and so forth. Solid materials, are they crystalline? Are they amorphous? Waxes, for example, might need a time afterwards to fully crystallize. If you are working with waxes and fats, what's their melting point, their glass transition temperature? And once you start to think about polymers, molecular weights. Often it's the highest molecular weight that gives you the best performance, but is the most difficult to work with. Whether you're melting it, it's highly viscous, or dissolving it, and you just can't dissolve a lot because it becomes too viscous. So properties to think about as you move forward. When we're thinking about the encapsulation processes, not enough time to go through all of the details, but just to give you some words to think about how you're going to atomize airflow rates, feed rates. You'll often hear batch versus continuous. So an emulsion-based process is typically batch. Spray drying is continuous. As long as you feed the spray dryer, it's going to keep making capsules. But for the emulsion-based processes, there's not always a continuous version available. You've made particles. How are they going to flow? Is it something flowable? Do you need something flowable? How are you going to make that? How are you going to collect? How are you going to filter? It's a puzzle. And that's where, as you start to think about all the different processes, seeking out help from experts who are either manufacturers, producers, research institutes, and so forth, to solve some of these combinations of materials and process. I mentioned earlier particle size. My preference is, how big can you go? How big can you accept? You're decreasing surface area, you're increasing wall thickness, increasing protection, mechanical properties. There's so many benefits to going as large as you can within reason, because of course there's going to be reasons why you want to go smaller, whether it's mouth feel, skin feel. Maybe you have to have it released quickly in a large capsule, just the surface area is too big. You're putting it into a beverage and you want it to look almost like it's disappeared. So you need something on the nanomaterial side, or you need something highly homogeneous in your final product. But if you're making maybe a granola bar, do you need something that's super monodispersed and small? No. It's crunchy. It's going to be heterogeneous. If you're going into a skin cream, yes, now you may need something smaller. And also think about distribution. It's always nice to want something narrow in size distribution, but that doesn't mean it's always better. And it's certainly probably not going to be cheaper in some cases. But there are great solutions out there if you do need to make some monodispersed capsules. In terms of payload or your shell thickness and capsule diameter. So payload, again, that's the amount of material we can put into a capsule. So a few examples here, you know, the egg, the yolk inside of the egg, the air inside of the balloon, the milk inside of the coconut. Here I have just a graph of your microcapsule diameter on the x-axis going up to 100 microns and shell thickness on the y-axis. 90% payload sounds great, 100 micron capsule, but your shell is going to be about 1.5 microns. That is very thin. Diffusion may be high for some environmental concern, water, oxygen, something like that. It may be brittle. And you're like, okay, it's 100 microns. Let's do a 10 micron shell thickness, right? 10 microns, that's about 10% of 100. That should be fine. Well, you got to remember your geometry and your mathematical equations here. So 10% shell thickness, that drops your payload to about 50%. So you've cut your payload in half to put a little bit thicker shell on there, which is great. Now you're gonna have a better capsule, but you're going to have to use twice as many capsules to deliver the same amount of active. So there's a trade-off. You have to do that cost-benefit analysis. And just a few examples. A latex balloon, I'm sure we've all played with water balloons. Those are not structurally robust. And if you let it sit out a while, they leak. You know, that water and that oxygen or that air comes out. But a coconut, you know, I'd rather be hit with a water balloon than a coconut. Coconut's not going to break. And it sits on the beach, it sits on a tree in the hot sun, and it still keeps moisture inside. It's also low surface area, but you know, the thicker the shell, the better. You, of course, have to think about how your capsule is going to release. This is part of the reason we encapsulate, is you're protecting, and then at some point you need it to release, or you need to protect it and just not have it release at all. So this absolutely has to go into your equation at the beginning before you start to think about encapsulation. You're like, I want to encapsulate because I need to protect, and then I need to release. And if you want it to release, what does that look like? You think about how quickly it might release, you know, what are some of the variables, things like diffusion, osmotic pressures, if there's something water soluble inside, what environment is your capsule going to be exposed to, not only in your final product, but during storage, shipping, processing, putting it into a consumer good. If it's going to mechanically rupture, what's the force and how are you going to measure it? That's a whole nother challenge. That's not as easy to measure as you know, taking a picture with an optical microscope. If there is a trigger, what's that trigger and what's the range? So there are pH sensitive capsules, but that pH sensitivity is more like I'm going from pH 3 to neutral. Everything's going to release. Great. Rather than I'm going from pH 3 to 3.1. That's not going to be a binary system of release there. It's going to be tight, tough to control release on that tighter range. So think about what range you have to work with, or temperature. There's some great solutions out there for color change and so forth, but temperature can be a challenging one. And then the release kinetics. So how quickly do you want your capsule to release? So here I have a graph on the lower side. You know, something like a matrix particle might be first order, because again, you have some material at the surface of your capsule. So that's going to pop off first. You're going to have what's called a burst release. And then the blueberries from within the muffin are going to diffuse out. The prettiest and most desirable release is zero order. You know, I want my capsule to start releasing and just be constant rate. That can be one of the most difficult to achieve. Core shell capsule is great at that, especially if your shell doesn't rupture, because then it's diffusion and you'll have a set rate in most cases across that shell. And then again, if you want that delay or trigger, say, I don't want my capsule to release until this trigger and then have it start to release. Plenty of release mechanisms, mechanical, that's your carbonless copy paper, some thermal release, permeation, shell dissolution. So that's a lot of your flavors, for example. Delayed release you see a lot in pharmaceuticals, maybe targeted. pH used a lot in pharmaceuticals and even some in agricultural applications. Osmotic, so if you have something water soluble on the inside, water will be sucked in and pop the capsule. That can be a nice burst. Biodegradation, photo and chemical. So plenty of ways, and there's probably some beyond this as well. So we've talked about what the capsule is, how to describe it, what you put in it, shell materials, how you make it, how do you analyze it? This can often be a major part of a project, is how you analyze the capsule. Our number one tool is microscopy. Look at the capsule. Now it's micro, so you're going to need a microscope. You know, cell phones are getting quite good and you can get away now with some rough estimates there, but a good microscope goes a long way. So a lot of the data that crosses our desk daily, optical microscopes, electron microscopy images, you start to get into particle size analysis. Now, a microscope can give you some particle size information, but if you really want a good statistical overview of the size, you're going to need laser diffraction, maybe dynamic light scattering if you're working with nanoencapsulation. Thermal analysis, very important if you're working with polymers and melt systems. Some way to look at the payload. That's going to be very specific. If I'm encapsulating a bacteria, that assay is going to look a lot different than if I'm encapsulating a flavor or fragrance. So you're looking at, pick your alphabet soup of chemical analytical technique, GC, HPLC, LC, NMR, FTIR, UV-Vis, and so forth. And then you get into your biological assays. How are you going to test release? Burst release, diffusion, controlled release. Now, pharmaceuticals has a lot of standardized methods, but you get outside of that industry and it's kind of the Wild West. You really have to think carefully about how you want to test, and your companies may already have some of those tests set up. Stability. So think about what that stability is, what kind of stability: suspension stability, microbial stability, thermal stability, moisture stability. If you say I want it to be stable, clarify what that is, over what time frame and what conditions, and then there may be some other assays you need to think about. So to wrap up here, at least give you our philosophy on capsule design, the way we don't do it and the way we recommend doing it. This is the wrong way. So this is you come up with a list of requirements chiseled in stone. They can't move. It has to be this size, this payload, this morphology, these materials, and we have to use this process. So, you stick that into the fictional encapsulator 5000 and you're setting yourself up for failure. There's no process, there's no, universal encapsulation process and formulation that will fit all categories. B, it's really hard to find something that magically fits all the criteria you can come up with in a brainstorming session. So if you're lucky, you hit the Holy Grail and you've got your solution, but 99.999 times out of 100 in terms of what we've seen, an immovable set of initial requirements is not going to work. So you have to be flexible. There has to be a strategic approach. So you pick a starting point between these five. And there's some other variables beyond this. These are the five main ones I'm focusing on here. So the size of the capsule, how much you want to put in the capsule, what morphology you're looking for, what materials you're going to use, what process you're going to use. So understand these are all related. You change one, it's going to change the other. So if I have spray drying, I'm probably not going to make a core shell microcapsule. Or if I want 90% payload, I'm probably not going to use spray drying. And so as you move these, we've got a spider web in the middle because it vibrates. Those changes will vibrate. So you pick your starting point, pick your anchor, and go through that feasibility exercise and balance those expectations in terms of what you're willing to give up and what you're willing to accept. All right, final comments. We are literally just scratching the surface. This is just the beginning. Again, 250,000 publications out there, 500,000 patent applications. There is a lot. I encourage you to know your limits. That's a big one when you're going and shopping and looking and thinking about making microcapsules. What particle size you're interested in? How do you want it to release? What do you want to protect it from? What are your material limits and how much do you need to put into this capsule? Think about these different processes. Each have restrictions in terms, are they batch, they continuous? Can I get tons per hour, tons per day, or am I making grams per hour and grams per day? And the cost, it's not a free system. So you have to pay for the materials, the equipment, the manpower. I haven't even touched on pre-processing. Maybe you have a powder you need to grind down or mill down, storage conditions and so forth. Also your industry. You're going to have regulations on materials. What are you putting the products into, the capsules into, and what sort of process? Do they go through a retort or a sterilization process? Is the encapsulation process you're looking for available? It's like, I really want to do fluid bed coating, but it has to be in this industry with this size material. And once you start looking at toll manufacturers, often there's certain restrictions that some have. Some only work in pharmaceutical space, some only work in industrial space, some only work with aqueous, maybe not solvents. So you think about availability and if you need to bring that process in-house on your own. That being said, with those limits, there's still plenty of opportunity. Really, the white spaces in these combinations of existing processes and new formulations. So don't be discouraged by the limits. There's still plenty out there that can be evaluated. So with that, if you do need any more information, certainly visit our website. Actually, we'll be putting some new information on there soon. We do have a 2.5-day in-person workshop that we offer twice a year. We've been doing that since 2016. And I've been teaching workshops like this since about 2005. Always in our workshop will be here in San Antonio. We have demonstrations of all the processes. We do welcome private visits and tours. There's plenty of expositions and trade shows as well to visit. There's other societies. I mentioned Controlled Release Society. There's also the Bioencapsulation Research Group. So they have several events a year. They have their own training workshop coming up later this spring in Loughborough in the UK. They also have industrial symposiums. Next one will be in Valencia this summer. If you have any questions, email me. And if for some reason you can't reach me via email, we have a general institute email address, ask@SwRI.org, and happy to answer any questions there. So with that, again, I thank you for your time. Thank you for your attention. Thank you for the opportunity to at least share some of our introductory knowledge on microencapsulation. Again, do keep in mind this is just an introduction, and happy to help and work with you and solve some of your encapsulation challenges. So thank you again.